Good morning, good afternoon, and good evening, everyone, from wherever you're joining us today, and welcome to the 8th webinar in the Neurophysiology 2023 series. Today's webinar is titled Systemic and Intraspinal Pathology and Repair After Spinal Cord Injury in Rodents, featuring Dr. Dana McTigue, Professor of Neuroscience at The Ohio State University. Today, she'll present her research on spinal cord injury models in rodents, including effects on other organs, metabolism, and myelination. Before we get started, we'd also like to acknowledge our partners for this series, the American Physiological Society, the Society for Neuroscience, the Federation of European Neuroscience Societies, the American Autonomic Society, and Scientist.com. We'd also like to give a specific mention to the Society for Neuroscience as the host society for today's event. We'd also like to say a big thank you to all of our series sponsors, in particular Alzet as the sponsor of today's webinar, as none of this would be possible without them. It's now my pleasure to welcome Dana to the floor.
Hi
All
there.
right.
Yep. Good to
Welcome,
go.
Dana. Yep, everything is ready to go. Take it away whenever you're ready.
Okay. Thank you, Andrew. And I want to thank everybody for joining today. It's a real honor to be part of this series. It's a great series, and I'm excited to go back and rewatch some of the other lectures that occurred already. And so today I'm going to talk about spinal cord injury repair and pathology using rodent models, as Andrew mentioned. And I'm going to mostly focus on intraspinal changes, but I will touch on some systemic issues that occur toward the end of the talk. So to begin with, a little history on me and why I got interested in spinal cord injury and the directions my lab has gone in. There's some data that came out back in 1993 from Dick Bunge's group at the Miami Project. And down there, they have a large tissue bank of spinal cord injured individuals who they were able to treat them while they were alive and then look at their spinal cord tissue after they passed away. And what they noticed, surprisingly, was that in 60% of the people, my laser on, 60% of people who were clinically complete, and that means they had no sensation and no motor function below the level of the injury, 60% of them were actually anatomically incomplete, meaning they did have some tissue spared at the injury site. So there was tissue remaining, but it clearly was not functioning properly for the individuals because they were having no function below the injury. And if you look at a typical human spinal cord injury, they often are contusion type injuries that occur due to car accidents or falls. And what happens is you get this large central region of damage at the epicenter area, and the lesion tends to spread rostral and caudally. But what you see around the rim of the tissue is a spared rim of white matter. And this is typical of almost all spinal cord contusion injuries. We have this tissue remaining, but oftentimes it doesn't function properly. And to me, early on, this suggested that it might be a problem with myelination. If we know that there are axons still remaining, but the axons aren't conducting properly, then perhaps there's an issue with the myelin along those axons. So when I started my laboratory, I used rodent models of human spinal cord injury that mimic this contusion type injury. So here's the injured contused spinal cord, and here's what it looks like when we do a comparable spinal contusion in rats or mice. We use a computer-controlled impact probe to do a quantifiable level of injury, so moderate, mild, or severe, at the impact site that we choose. This leads to the central region of hemorrhagic necrosis, shown here in this blown-out 3D image, and then some lesion extensions, both rostrally and caudally, in the spinal cord. So our rodent models very nicely mimic the contusion-type model that happens in the majority of human spinal cord injuries. So within this model, when I started my laboratory, being interested in myelination changes, I began to study the oligodendrocyte lineage. And this is what happens normally during development. We'll have stem cells that go down an oligodendrocyte pathway. These progenitor cells, are also called polydendrites or OPCs, tend to express some different developmental markers like NG2 and PDGF receptor alpha. And you'll hear me refer to these cells often as NG2 cells throughout the talk. These NG2 cells, then during development, some of them will go on to differentiate into premyelinating oligodendrocytes, and they downregulate their markers like NG2 and PDGF receptor alpha, start to upregulate some oligodendrocyte markers. Then as these cells find axons and form mature myelin, it's at this point that they're considered a myelinating mature oligodendrocyte. So this is what typically happens during development. But it turns out there's a population of these NG2 progenitor cells that are maintained into the adulthood. So these NG2 positive cells have these long branch processes They're actually found throughout the white matter and the gray matter in the adult central nervous system, throughout the spinal cord and the brain. And for a long time, when I first started my lab, gosh, now over 20 years ago, there wasn't as much known about these cells. They were still relatively new. And so when my lab started out, I wanted to understand, what do these progenitor cells do in the injured spinal cord? We knew that in MS models, these cells can divide and they can migrate to lesions, they can form new oligodendrocytes, and they can remyelinate axons. So I wanted to find out, do they have a similar role after spinal cord injury in the adult injured tissue? So one of the first studies I did with my first student, Richard Tripathi, was just to look to see, do these cells survive injury? Do they form new oligodendrocytes? And if so, where do these occur and when is it happening? And so Richard did a short time course study looking at NG2 cells and then looking to see if we can find evidence of new oligodendrocytes. And to find those, we used a marker called CC1, which is nice because it labels oligodendrocyte cell bodies, but not all the myelin. So it makes it easy to find the cells. And then we also give the animals BRDU that would label newly divided cells or newly formed cells after injury. And what Richard found out was that if we look compared to normal white matter shown in the dotted line, quickly after injury, there's a huge drop in oligodendrocyte numbers at the lesion epicenter, as well as extending both rostrally and caudally. And then over time, over the first 2 weeks, these cells are replaced, apparently, and they rebound back to almost normal levels. And in this image, you can see cells labeled for oligodendrocytes in brown and astrocytes in black. Interestingly, along the lesion border, so that the area that's right along the lesion, so this would be that contusion area above, this is spared tissue. This lesion border had massive amounts of new oligodendrocytes. So not only did they not really drop that much by 3 days post-injury, over the next 2 weeks, the NG2 cells divided and differentiated to such an extent that we had significantly more oligodendrocytes along this lesion border compared to before injury. And you can see that here with all this cluster of brown cells along the lesion border within this astrocyte scar. So oligodendrocytes do seem to be replaced after injury, at least acutely, and this seems to be a very robust reparative response. So some of the next questions we wanted to find out was, how long does this last? And do these oligos do anything? So do they actually remyelinate axons? So to answer those questions, we moved on to some mouse reporter models. And this was work done by one of my former students, Dr. Zoe Hesp, shown here on the right. And so at this point in time, we had reporter mice where we could take our oligodendrocyte progenitor cells and turn on GFP in the cells so that they would become marked with green fluorescent protein. And then if they differentiated into oligodendrocytes, the oligos would also be GFP positive. So what we did with the model was we took mice, we also used a rat model as well and saw similar results. So we do a spinal cord injury in the mid-thoracic level. We could give the animals tamoxifen at some point in time, and the tamoxifen then would turn on the GFP in the OPCs. Then if that cell went on to differentiate, we could see new green positive oligodendrocytes. So Zoe first started out by giving tamoxifen early after injury, since we knew that this was a time of robust oligogenesis. And we wanted to see if we turn our OPCs green at the time of injury or just a couple weeks later, and we give them a couple weeks to divide and differentiate, can we see evidence of new oligodendrocytes? And the answer was yes. We could find evidence of very nice, beautifully labeled GFP positive oligodendrocytes throughout the tissue around the lesion border and even extending a bit rostrally and caudally. So we saw in this early time period, we did see robust oligogenesis, and it seemed to be ramping up. So we wanted to go on and look at some more later time points. And for this, Zoe waited she did the injuries and then didn't give the tamoxifen until week 6 or even week 8. So the beginning of the 3rd month post-injury, we turned our OPCs green and then waited a couple weeks to see if we could find evidence of green oligodendrocytes. And since I'm telling you this, the answer was probably yes. So we did see this. So when we turned the OPCs green at 6 weeks or even at 8 weeks post-injury, we could still find evidence of GFP positive CC1 cells indicative of new oligodendrocytes. You can see here in this spared white matter in the longitudinal horizontal section, these oligos with processes that look like they're myelinating axons potentially. And we found this again in the white matter as well as in the gray matter in spared tissue. So this shows us that oligodendrocyte genesis is surprisingly a long-lasting phenomenon after spinal cord injury. And when I first was trained in spinal cord injury, we were kind of taught that the injury occurs, you have this response over the first couple weeks, and then things stabilize, and you don't get a lot of changes after that. So we actually thought in this, the GFP 8-week study that we wouldn't really see much changes, but we did see evidence still of new oligodendrocytes being formed. So we put this data all together. What we actually saw was that it didn't seem to matter when we gave the tamoxifen. OPCs were making oligodendrocytes within the first week, as well as some of them waiting until the 10th and 11th and 12th week to make oligodendrocytes. So we have some OPCs that are dividing right away and differentiating. Meanwhile, others are hanging out for several weeks to months before they then go on to make a new oligodendrocyte. We wanted to know then also, what are these oligos doing? So are they myelinating axons? Because just because they're there doesn't mean they're actually doing what we're hoping they're doing. And so we did some double label confocal microscopy and 3D, confocal Z-stack imaging, and we're able to find lots of examples of axons, as an example here stained in red, with GFP surrounding the axon. So what looks like axons being myelinated by this oligodendrocyte. And in this image, you can see a Z-stack through a spinal cord axon. Here the axon is labeled in blue, and MBP is labeled in red, and then we have the GFP as part of this MBP positive sheath. So we could see what looks like evidence of myelination. This doesn't prove it, however. So in order to try to find more evidence that this is actually functional myelin, we turn to examining the nodes of Ranvier. So if you're familiar with the node of Ranvier, you'll know that nodes are actually highly complex structures. There are a lot of different connections between the axons and the myelin that are holding the different profiles and different proteins in place. And a node of Ranvier doesn't form unless there is intact, compact, functional myelin. And this is because the myelin membrane at these perinodal loops actually connects with axon proteins called Casper. And this Casper myelin connection is what helps them to keep the nodes organized. And the organization of the node has the sodium channels right here in the middle where the action potential will be taking place. We have this perinodal region that's formed by Casper and other molecules. And then adjacent to that is where the potassium channels are. So the Kv1.2 channels are just under the end of the myelin. This keeps everything nicely organized and helps to keep the saltatory conduction flowing in a proper way. So if we were able to look into our spinal cord tissue and find evidence of intact nodes where we see the green myelin, that would indicate that the myelin was functional. And in fact, we did see that. So we saw lots of examples of axons like this one here labeled in blue with GFP surrounding it. And then this GFP is ending in Casper positive profiles, which is indicative of a node of Ranvier. And so what we think happens with this axon is this side was myelinated by new myelin. This side is GFP negative, meaning that it must be spared myelin on this side. It's probably not unmyelinated because otherwise then the node wouldn't have formed properly. So this was evidence, an example of indicating that this myelin that the new oligos were forming seems to be functional. We also did some immuno-EM, to show that we can find evidence of GFP positive signal within myelin wraps, shown on the EM. So all of this indicated that these new oligodendrocytes actually are able to repair myelin after spinal cord injury, and that we saw this out through at least 2 to 3 months post-injury. And this also was interesting because there is some evidence from the 70s and the 80s using EM to look at spared myelin, or look at spared white matter in spinal cord injury models in cats and in rats. And they were able to show evidence of remyelination occurring after injury, but it seemed to be mostly occurring, what they saw was around 3 to 4 weeks post-injury. They could see evidence of new myelin forming. And so what we're seeing here in this model using our reporter mice is that this new myelin appears to be formed for several months. However, we couldn't really quantify the amount of new myelin happening in these animals because this particular mouse model would make not only the new oligos GFP positive, but also our OPCs were GFP positive. So we couldn't just measure the amount of GFP as an indicator of myelin, because as you can see here in these images, these are some OPCs labeled with NG2, right, that are also double labeled with GFP. So that means these cells got recombined and expressed the GFP, but they did not differentiate into oligodendrocytes or myelinate. So some of the OPCs do differentiate, whereas some nearby OPCs do not. And why some do and why some don't, we don't know yet. but so we couldn't really measure or quantify how much new myelin is being formed over time after spinal cord injury. So to get at that question, one of my more recent students, Dr. Nikki Pukos, who graduated just a couple years ago, did a study with a new reporter mouse that we got from Bill Richardson at University College London. And in these new mice, we could again give tamoxifen to the animals at a certain time post-injury, and the OPCs would be affected by the tamoxifen, but they would not express membrane-bound GFP unless they differentiated into new oligodendrocytes. So in this mouse model, we were able to cut out the GFP-positive OPCs and know that if we see GFP-positive cells or myelin, that is all from an oligodendrocyte and is not confounded by the OPC labeling. So in this model, Nikki again did mild, moderate contusions in the mid-thoracic level of the mice spinal cords, and then waited it to different times to give the animals tamoxifen. So for this example here, some of the mice got tamoxifen at the beginning of the second month post-injury, and then we perfused them at the end of the second month to look at myelin changes at this point. Some mice did not get tamoxifen until 3 months post-injury, and some we figured we'd wait for a long time and see, if we wait out to 6 months post-injury, we would anticipate that there will not be much going on at that point in time. So she gave tamoxifen at the beginning of the 6 month post-injury and then stopped at the end and looked for evidence of new myelin, new oligodendrocytes. What we were able to show is that, in fact, we did find evidence of new myelin being formed at every time point that we looked. So compared to age-matched naive animals who do have a low level of new myelin forming, there's always a little bit of new myelin formation going on in the central nervous system. The animals that received spinal cord injury had a significant increase in the amount of new myelin formed. So these images are longitudinal sections of white matter from outside the lesion, either close to the lesion border or distal white matter. And you can see the GFP positive label here in green. These sections were also double labeled for axons. So we were able to quantify the amount of neurofilament labeling that was double labeled for GFP positive myelin. To ensure that this myelin was functional, then we also looked for node of Rambier formation. And as you can see from these images, this is an example of a GFP positive axon or GFP positive profile here. It looks like it's running along an axon profile. And both GFP ends of the myelin are ending in Casper positive profiles. So indicative of a mature node of Rambier that was formed by this new myelin. So this is a high power example and some lower power examples over here, showing that we could find evidence that the myelin appears to be functional after formation after spinal cord injury. To get at the quantification, then Nikki went to we used a program called MyPAR to analyze the amount of axons that were double labeled with new myelin. And she was able to show, based on the images, that while we do see new myelin formed at each time point that we examined, the most myelin was formed during this third month post-injury. So animals, when she looked at it 2 months post-injury, had significantly more myelin, shown here in the green line, than the animals that were from this 3 month post-injury point. You can see had significantly more myelin compared to the earlier time points and compared to age-matched controls. And then with the 6 month animals shown here in pink. So we still see evidence of new myelin, quantifiably increased new myelin compared to uninjured animals at the 6 month post-injury. Interestingly, also, this new myelin, while it was closest at the epicenter region, it extended several segments rostral and caudal. So in this lesion model, Nikki looked at the epicenter at the 0.5, plus or minus 0.5 millimeters, but out to 2 millimeters rostral and caudal is quite distal from the lesion, and it's not in the lesion proper. So even out in what looks like spare tissue, we're seeing evidence of new myelin formation. She was able to also show that this new myelin is accumulating over time. So one question could have been, did this new myelin that was formed early, was it somehow damaged and lost? And some of the later myelin then is just replacing this myelin. This was a question that we've gotten different times in our data in the past. So to look at that, we look to see, do the oligodendrocytes accumulate over time? And does the myelin accumulate over time? So for some of the animals, she took animals at 4 weeks post-injury and looked at the number of oligodendrocytes. So the number of total oligos, the number of new oligodendrocytes was significantly higher than the uninjured animals. So again, replicating what we saw before, that oligogenesis is actually very robust after spinal cord injury, leading to levels above normal. But then when she looked at animals, she compared the 4 weeks to the 10 weeks post-injury. There were significantly more oligodendrocytes, more new oligos, and more new myelin. So this suggests that these oligos and this myelin is continually being added over time after spinal cord injury. So to me, this data was really exciting because it shows that the spinal cord is not stabilized as I was taught as a postdoc. It's not, just quiet and nothing else is going on. Actually, for 6 months post-injury, the spinal cord is working to repair itself. It's forming new oligodendrocytes, forming new myelin, and it's actually quite dynamic over time. So the injury site is highly dynamic, and we can now see that these endogenous reparative responses continue for several months after injury. I think this is exciting because it shows the tissue is still in a responsive state, that it might be, responsive to manipulation to try to increase, the function even beyond what happens spontaneously. This data also led to a question that if there's chronic repair, what is being repaired? So if we're getting new myelin being formed and it's building up over time, what is being myelinated? There must be axons in there in need of myelination. So chronic remyelination suggests that there must be some chronic demyelination. There'd be demyelination, or there could be new axons being formed that need to be myelinated. And we ruled that out in our studies. So the next thing we wanted to look at to see was, is there chronic demyelination in our animal models? There has been some anecdotal evidence in human tissue that there are bare axons, especially along the lesion border, in human spinal cord injured tissue samples. And so, but it hasn't been looked at quantifiably carefully in animal models for a long time. So we wanted to then look to see, can we find evidence of chronic demyelination in our injured spinal cords? And the way, like I mentioned earlier, people typically do this is with electron microscopy. So this is an example from a paper from Jean Rathal's group, where she looked in rat spinal cord injuries at 2 months post-injury using EM, and you can see these green arrows are indicating demyelinated axons. So this really shows there is some demyelination there chronically. You can see in contrast to these big, nice, beautiful myelin wraps. These are the spared myelin axons, and the thin myelin actually is indicative of remyelination. So in this one field, they can see evidence of axons that were spared, myelin that was new, and axons that never became remyelinated. They remained demyelinated after injury. So this is a really beautiful way to look for demyelination. However, it takes dedicated animals to do this tissue prep. There's special prep needed for this tissue, so you can't really use the tissue for much of anything else. There's a lot of toxic chemicals involved in preparing this tissue, and it takes a long time and special expertise to get the results. So we do this in our laboratory. We also wanted to see if we could find a way to look more easily in the tissue we had available in our reporter mice tissue that we stained for the GFP, to then also look to see if we can find evidence of demyelination in our tissue sections. And so to do that, we turned again back to the node of Rambier analysis. And like I said before, the myelin keeps these nodal proteins highly organized. What happens when there's demyelination? These nodal proteins actually spread. So they kind of tend to get lost. And you can see here's an example of sodium channels and potassium channels that used to be highly organized and separated. They spread along the lesion, these axons, to become intermingled. And as you would know, then this would no longer allow saltatory conduction and rapid axon potential conduction. So we wanted to see, could we find evidence of nodal protein spread in our tissue sections that would be indicative of demyelination? And it turns out this has been examined at least at one time point by Michael Fahling's lab from Canada. And in his tissue, they looked at potassium channels, shown here in green, and double-labeled spinal cord injured mouse tissue with neurofilaments in red. And they can see in uninjured tissue, you can see evidence of what you would expect, these nodal clusters of potassium channels. But in the animals that had spinal cord injury, look at these potassium channels, they're all over the place. So they've spread all out up and down along the axons, which would be indicative of demyelination or myelin loss. We wanted to look then in our animals at the different time points that we've shown for the remyelination to occur. So we took our tissue sections and stained them for KB1.2 in green, which is the potassium channel, and then Casper, which is the channel that connects to the myelin, which is right inside the potassium channels. So we double labeled our tissue sections, and you can see evidence of these really beautiful little nodes. So these are the Casper doublets that are surrounded by KB1.2 channels. And then in this image, this section is double labeled or triple labeled for axons shown in blue. So here's a really nice example of KB1.2, Casper, Casper, KB1.2, indicating a normal node. And this example is actually from one of our injured mouse tissue sections that is in white matter quite distal to the lesion epicenter. If we look in the same section now close to the epicenter, this is what the tissue looks like. The potassium channels you can see are spread for long distances along the axons. Casper channels are also tend to be spread. Casper proteins are spread and disorganized. And even there's examples of this overlap. So here's an example. You can see Casper and potassium channels completely on top of each other, right? So shown here in these white profiles. So this suggests and shows to us that at least in this spinal cord, which was a 7-day post-injury cord, we do see evidence of what looks like demyelination along spared axons along the lesion border. So what Nikki Pukos did then was look to see if she could quantify the nodal protein changes after injury as an indication of demyelination. And so she was able to show in our normal animals, a normal node, these profiles typically are around 5 microns in length. So the potassium channels are 5 microns and the Casper segments are also right around 5 microns. So we were able to use our online image analysis system to quantify profiles that were within this range or profiles that were significantly longer, like you would see on this example right here with this spread along the axon. So Nikki looked at there are different time points and was able to show at 1 week post-injury, you can see there's beginning of spread here. These potassium channels are getting longer. And then over time, this continues. Even out to our 6-month post-injury tissue sections, here's an axon labeled in blue. And look, these potassium channels are all along the axon. And there are also some Casper channels or Casper proteins mixed in, as you can see here. So we see evidence of longer profiles and shorter profiles, overlapping profiles, all indicative of axons that are still there, but their nodal proteins are not organized in the way that would suggest they're able to fire properly because of myelination. So quantifying this is what the graphs look like. So it's a lot of data, but you can see again, here are the epicenter sections, which correspond to around this area in our mice spinal cords, out to 2 millimeters rostral and caudal. And these bars are showing how these tissue sections change over time. With our naive animals low, so this graph, I should say, is showing you the elongated potassium channels. So in this graph, we imaged and quantified how many of these channel profiles are longer than 5 microns, like these shown over here in this example. So we can see in naive animals, very few are that long as we expected. Within 7 days, shown in the pink bars, we see a significant increase in these long channels. And this is maintained out through at least 6 months post-injury, shown here in the light blue bars at the end. So there's evidence that even though there's this remyelination going on, at the same time, we do have evidence still of demyelination and nodal spreading. We wanted to also use another way to quantify demyelination. You always want to try to find as many different ways to confirm your results as possible. So we turned to another thing that's been known in the MS field, and that is that when there's demyelination in the adult brain and spinal cord, the sodium channels actually switch to one that's present in the developing brain and spinal cord. So the adult CNS expresses sodium 1.6 channels, so NAD 1.6 type channels, but during development, there actually is a subtype called 1.2 sodium channels. As animals develop and myelin is formed, these channels switch to 1.6. So in the adult CNS, you only see the 1.6 channels. It's been shown in MS tissue and in MS animal model tissue that you can see an upregulation of these NAD 1.2 channels. So these early, less efficient channels become upregulated after demyelination. So we again wanted to see, is this happening in our spinal cord injured animals? This hadn't been examined before. So we stained our tissue for NAD 1.2, and as you can see here in the naive animals, this is longitudinal spinal cord white matter section, really no evidence of NAD 1.2 expression at all. And then within 1 week, we start to see upregulation of the sodium channel that seems to continue over time, at least through 10 weeks post-injury. Again, you can see these long profiles. They would be double labeled with neurofilament if we show the neurofilament channel. So these axons are upregulating this immature sodium subtype, and it seems to be actually somewhat decreased by 6 weeks or 6 months post-injury. So, and that's shown here in the quantification. So we have in our naive animals, you don't even see the bars because there's no NAB 1.2 in that tissue. This sodium channel upregulation seems to ramp up through 28 days post-injury, and then it's declining actually significantly at 6 months post-injury. So to me, this suggests that perhaps this new myelin is helping to start to restore the axons back to a potentially more normal phenotype of their nodes and their sodium channels, but it's obviously not completely back to normal since we still do see evidence of nodal spreading and some NAD 1.2 expression. Putting this together, what we see then, what this data tends to show, is that over time after injury, we do have this long-term ongoing spontaneous repair process, but there seems also still to be some injury process there that is either the demyelination is ongoing, and it seems to be that it is ongoing because if we look back at these data, it seems like this is ramping up over time. So it's possible that some axons that initially had spared myelin are becoming demyelinated over time. So indicating that there might be this kind of this cycle that's going on all the time. We have some injured tissue, we have loss of myelin, the cells come in, they try to repair that, but then there might be some more loss of other myelin, and this is an ongoing process out to at least 6 months post-injury. So again, the tissue is very dynamic for long periods of time. One of the things we've been starting to ask now in the lab is what's driving this? So what are some of the mechanisms that are driving some of this reparative process? If we can understand potentially what the cells can respond to, we might be able to then push the system more toward the repair side of the cycle and alleviate some of the injured side of the cycle. So for this, Nikki turned again to some really cool data that had been coming out in the MS field, showing that axons can regulate their myelination by releasing glutamate. And so axons actually do not only release glutamate at the terminals, they actually release glutamate along their axon bodies as well. And this occurs typically on axons that are not myelinated. And interestingly, axons that are not myelinated tend to have these OPCs putting processes right along these axons. And the OPC processes abut to axons where there's V-GLUT2 expression. So V-GLUT2 is a glutamate transporter, and this is indicative of an area where an axon is going to release its glutamate. And this paper went on to show that after demyelination, this association between NG2 cells and V-GLUT puncta increases, that if you stimulate the axons, they release glutamate, that the OPCs respond to the glutamate, the OPCs express AMPA receptors, and that this can then lead to increased myelination in this demyelinated model. So, and if they block the glutamate release, it reduced the remyelination. So suggesting that a really cool thing that's happening in the central nervous system along axons is that axons are releasing their glutamate. The OPCs are listening into that signal, and when they get the right signal, the OPCs then can differentiate into oligodendrocytes and myelinate those axons. So Nikki then wanted to see if this happens in our traumatically injured tissue. It hadn't been examined before. And so what she wanted to find out is, do the OPCs in the spinal cord contact glutamatergic axons like it was shown in the brain? Do they do this normally or after injury? And if so, does glutamate play a role in some of these OPC responses? So the first thing we did was look for the V-GLUT puncta in the white matter after spinal cord injury. And Nikki showed that, interestingly, the V-GLUT puncta number increases over time. So this is per axon profile. So we know that there may be we know there are axons that are lost after injury, but if we normalize for that, of the axons that are still there, they're increasing their number of V-GLUT puncta along the axon. So these are little spots where the glutamate can be released from these axons in the white matter. She did double labeling for NG2, then to see if the OPCs are touching the axons where the V-GLUT puncta are located, and the answer was yes. So these happen normally in the normal uninjured tissue, and then this also increases over time and is maintained for 6 months post-injury. So the NG2 cells are abutting these axons, and using our image analysis system, she was able to quantify regions where they're double labeled. So the OPCs are increasing and seem to be listening in to the glutamatergic axons in the injured spinal cord. To see if the glutamate is actually playing a role in the OPC responses, we turn to DREAD technology. So designer receptors exclusively activated by designer drugs. As hopefully most of you are familiar with now, you can use viruses to express these DREAD molecules in specific cell types and then give the animal CNO that will either activate or inhibit those channels, enabling you to activate or inhibit specific neurons and axon signaling. So Nikki used viruses to express inhibitory drugs, first of all, in the rat spinal cord, and used a virus in which we knew that they would be expressed only in glutamatergic neurons within the spinal cord. And these viruses also express mCherry. So we're able to see in our animals the axons that were actually from neurons that had received the DREAD signaling and were upregulating the DREAD receptors. So when she took these animals and gave them CNO, she was able to show that the number of NG2 contacts on the silenced axons was cut in half. So there were significantly reduced NG2 axon contacts when the axons had been silenced by the CNO. So this is really cool because it shows that spontaneously these NG2 cells and axons are interacting after spinal cord injury. The NG2 cells are listening to the axons are active, and this activation is helping to drive this cellular interaction. We did the opposite, where we increased activation of these axons using an excitatory DREAD. Again, the DREAD also expressed mCherry. Here you can see the axons that are positively expressing mCherry and the excitatory DREAD after injury. And when we gave these animals CNO compared to the control, there was a significant increase in the number of OPC cells contacting the axons. So again, this shows that not only are the OPCs responsive to the spontaneous activity, they can be driven even further to contact the axons by increasing axon activity. These data, we're still, working on some of this tissue. This data was just published recently. We know that within this study, this was a short time point study, so we did not see changes in the number of oligodendrocytes or myelination because we didn't let the animals go as long for that. So those studies are ongoing now to see if we're actually getting not only the interaction between the cells and axons, actually getting evidence of oligogenesis and myelin repair. And especially if we can drive that even further and perhaps then reduce some of the long-term loss of myelin. So putting this together, this just shows what we've shown collectively in this study so far. We have spinal cord injury that seems to go along. If we look in the long term after injury, we can see evidence of extended examples of demyelination. So we see disorganized nodes, we see longer sodium channel and potassium channel profiles. We see sodium channel switching to an immature phenotype. But at the same time, we also see evidence of long-term new myelin being formed throughout 6 months. So we see these new myelin segments forming along some of these axons that probably lost their myelin. And then we know that we can take this process and drive it further by increasing axon activity. So I find this really interesting and exciting because this shows that the chronic spinal cord, so this is 6 months in a mouse. So I don't know what that would equate to in a human, but obviously much longer than 6 months. So it suggests that this tissue is remaining amenable to interactions and to interventions that perhaps could be used to promote some tissue repair. And if you've been following any of the spinal cord injury literature, you would know that there's a lot of excitement right now about epidural stimulation, and they are taking the spinal cord injured individuals doing epidural stimulation, which is going to activate axons, and they're seeing some remarkable recovery in individuals who've been chronically injured for years and had no level of recovery. So it's really exciting to think that one of the mechanisms they may be tapping into is this remyelination mechanism. So as I mentioned earlier and in the introduction, we talked about the fact that my lab looks at intraspinal effects, but we also have begun looking at more systemic effects after spinal cord injury. So I'm going to switch now from talking about what goes on within the spinal cord and just briefly cover some of the data that we're looking at in the systemic effects of spinal cord injury. And it took the field a while, I think, to actually get to looking at the rest of the body. There's been, years and years of work looking at the spinal cord itself, trying to understand what happens in the pathology there, how to repair the axons in the lesion, and not really thinking about the fact that the spinal cord actually innervates the entire body, innervates all of our organs. And so if the spinal cord is not functioning properly, it's highly likely that a lot of the organs, again, also are not functioning properly. So I'm always asked, how did I get from looking at oligodendrocytes, looking at metabolic phenotypes, and especially what I'm going to talk today about is liver phenotypes. And the reason we looked at this, there's a logical progression, and for evidence, for thoughts for the students out there, this data really showed to me that you never know where your career is going to take you, and you can end up going into some really interesting directions. So in my lab at one point in time, we were looking at iron regulation. Iron is needed to make new myelin. It's needed by oligodendrocytes. It's an essential component of the catalysts that activate the enzymes. So we know that oligos need iron, but after spinal cord injury, there's bleeding within the spinal cord. This leads to iron overload. So I had some studies looking at whether or not we could give an iron chelator to dampen down some of this iron overload, but also not inhibit or impair remyelination. So in these studies with former postdoc Andrew Sauerbach, gave our animals a systemic iron chelator, and we were looking in the spinal cord for changes in tissue sparing and tissue repair. But Andrew had the good idea to look at the liver just as a proof of concept, because the liver is the highest iron containing organ in the body. And if this chelator was working, we should see reduced iron within the liver. So Andrew looked at the livers of some of his animals that he'd given the chelator to. He noticed that the livers, compared to the uninjured animals, they didn't look right. They looked pathological. They looked inflamed. And so this was like what is going on here? We know from other studies that have looked acutely after spinal cord injury that there is evidence that livers become inflamed right after injury. And this is typical of any kind of major injury. The livers respond with an acute phase response, which has the goal of promoting inflammation, inflammatory cytokines, mobilizing immune cells, promoting clotting, things like that. Things are going to help repair whatever the wound is or the injury that happened. So we knew the livers became somewhat inflamed after injury. But what Andrew was showing now in his chronically injured animals that were months after injury, we noticed that they seemed to be pathological and they still were showing evidence of inflammation. So I wanted to follow up on this a little bit more. We know the liver is like one of the most important organs in the body. It's the largest organ in the body. It's important for a plethora of functions in our body. And if the liver is not functioning properly, that could be a major problem for individuals with spinal cord injury. So we decided to initially do a time course study to look at the liver after injury to see when are things changing and how long is it lasting. And so we looked at liver inflammation and a bunch of different outcomes. And what we saw, I'm going to show you just some data, some collective data from our different studies. The first thing we looked at were Kupffer cells. So Kupffer cells are the liver macrophages. These cells live in the liver. This is actually the largest population of macrophages in the body. They occur within the liver. These Kupffer cells line the hepatocyte sinusoids, the liver sinusoids. And what we were able to show is that after spinal cord injury, these Kupffer cells, labeled here in black, you can see there's significantly more of them, and they're significantly larger. So these Kupffer cells become activated after spinal cord injury within the liver, and out to 6 months post-injury, they did not calm down. So the liver macrophages are mobilized and activated rapidly, and then they never receive the signal then that things are back to normal and they should go, they can stop being so reactive. Accordingly, we saw elevated cytokines within the livers. So if we did PCR for pro-inflammatory cytokines, you can see TNF is up early and is maintained. Here we looked out to 8 weeks post-injury. Same with IL-1 alpha and IL-1 beta. So these cytokines are also significantly increased, showing that the livers really do maintain a high level of inflammation after spinal cord injury. We looked also at lipid accumulation. The liver, one of its functions is to store lipids in the body, and you can see in this naive animal, oil red O labeling, which labels lipid droplets, you can see some lipid droplets as you would expect. But after injury, these seem to be significantly increased. And this is relevant because elevated lipids can become cytotoxic and damage the nearby hepatocytes, especially if they're present in a situation of inflammation. And to look to see if we are having liver damage, we looked for serum ALT levels in our animals. And ALT is actually a liver function test you get whenever you go to the doctor's office and they want to see, they measure your liver enzymes to look for liver damage. What they're looking for is serum ALT. ALT is a protein from hepatocytes that leaks out when the hepatocytes are damaged. So we were able to see in our animals that just got spinal cord injury, over time, their serum ALT levels were significantly increasing, indicating that the livers were actually becoming pathological and damaged after spinal cord injury, and that this was lasting for quite a while. So what is the significance of these particular findings? This actually, when livers have elevated inflammation and lipid accumulation, it's a disease called fatty liver disease or non-alcoholic steatohepatitis. So fatty liver disease is typically known to occur in individuals with high-fat diets and obesity. But our animals were not on a high-fat diet. They were lean animals, but yet they still showed evidence of this advanced form of fatty liver disease called non-alcoholic steatohepatitis. Why is this important? So this is important because this condition, also known as NASH for short, NASH is known to promote insulin resistance throughout the body. Insulin resistance is the precursor to diabetes. NASH is also an independent risk factor for cardiovascular disease. So people who have NASH have a much higher incidence of heart attacks and strokes. NASH is also a precursor to hepatic carcinoma or liver cancer. And it turns out that the spinal cord injured population have significantly increased incidences of all of these different conditions. So we know in spinal cord injured people, diabetes is 3 times higher than in the able-bodied individual population. Dyslipidemia or elevated blood lipids is 6 times higher. Heart attacks occur at 8.5 times higher. Not only do these occur at higher levels, they occur earlier in life, and they tend to be more severe in the SCI population. We also, cirrhosis and liver cancer are significantly higher in individuals with spinal cord injury. So our data are showing that our animal model is ending up looking a lot like an animal model of obesity. And we've done some newer studies in the lab comparing what these indicators, what these look like in obese animals versus spinal cord injured animals. And surprisingly, spinal cord injury is just as severe as obesity, and in some cases more severe than obesity for some of these outcomes. So look for that data coming out soon, hopefully, and more data we're following up on this within the lab. So putting this all together, wrapping up here, what I've shown you today is that we have spinal cord injury occurring in the adult nervous system. We have ongoing repair and injury within the spinal cord itself, and then this injury is setting off what we know to be hepatic inflammation and pathology. Other evidence in the lab and other people's labs have shown this also helps to drive systemic inflammation. Collectively, this leads to what's called metabolic syndrome, which I'm sure you've heard about in terms of obesity. This metabolic syndrome promotes obesity, diabetes, cardiovascular disease, and leads to increased mortality. And the SCI population has increased mortality. Their survival rate has not increased in the last 30 years. But there's a lot of hope now that people in the field are focusing a lot more on some of these systemic issues, in addition to the spinal cord, to try to improve their health and their lifespan and their quality of life. So with that, I will end and thank my laboratory. This is our lab picture from last year, our Christmas party at an axe throwing event, which was very fun. So I have an awesome laboratory filled with brilliant postdocs and students, former laboratory members listed here who helped with some of the work I showed today. My Ohio State colleagues, I live and work in a very intellectually rich environment here at Ohio State with lots of people with varied expertise, which contribute to the studies. And then also, of course, my funding. I need to thank all the funders, including the Belford Family Foundation. So with that, I'll be happy to end and take any questions you may have. Thanks.
Okay, so ready to answer some questions?
Sure.
All right, I'll start with a couple of simple ones, just procedural here. What was the sex of the rodents used in the study with tamoxifen?
They were both, actually. We used male and female, and we didn't see differences in the different sexes in the responses.
Okay.
So we use all of our animals.
Okay, great. Did you have a group of SCI rodents that tamoxifen wasn't used in? And if so, was there any difference in oligodendrocyte formation?
We did. Yeah, actually, my student Nikki has a whole paper on that, looking at tamoxifen effects alone and looking to see if that affects myelination or oligogenesis. And there was no effect. She looked at multiple different outcome measures. So if you just give tamoxifen alone, which is a really great question, that does not seem to be affecting what the responses that we're looking at.
Okay, fantastic. How long and how frequently do you need to treat with CNO to see the effects of altering V-GluT2 neuron activity on the number of NG2 contacts?
Yep, we gave the CNO for 7 days, and then we looked 2 weeks later.
Okay.
So.
This is a great question, a bit speculative perhaps. Are the same processes going on in peripheral nervous system injured axons?
That's a really good question. I don't suspect they're going to be identical because the peripheral nervous system has Schwann cells, and the Schwann cells act very differently from the OPCs. The Schwann cells themselves can dedifferentiate and help to promote axon regrowth. But I do know some of the growth factors are similar, and the inflammatory cytokines are kind of similar in the CNS and the PNS. So there probably could be some overlap, but I bet they're not identical, would be my guess.
Okay, great answer. Yeah, to be seen, I suppose.
Right.
How can this question is probably on a therapeutic, on the
Mhm.
therapeutic side. How can the remyelination potential be tapped into to improve recovery from spinal cord injury?
Yeah, that's a great question. So one thing I think is, like we mentioned with the epidural stimulation, that's going to be activating axons, so that may tap into it. Another thing we're looking at in the lab and others have been looking at is activity-based training. So we have animals in the lab. I have a former study with a colleague, Michelle Basso, where we showed if we put spinal cord injured mice on a treadmill, there was increased new oligodendrocytes formed, interestingly, in the cervical spinal cord even. So the animals had a thoracic injury. They were on the treadmill, so their forelimbs were getting increased activity. And in that part of the spinal cord, we saw more new oligos being formed. Exercise increases growth factor expression within the CNS, and the OPCs are highly responsive to multiple different growth factors. So we think that exercise may be another way to tap into it. Then, of course, I'm sure there are different therapeutics we could try to tap into as well. So we have some work going on now doing like proteomics and transcriptomics to look at changes in receptor expression and things in the OPCs to try to understand what exactly they'll be responsive to over time.
So understand the process as a first step, and then be able to tweak and
Right.
enhance certain elements of it, different approaches. Fantastic.
Yeah.
Here's a great question. What sort of mechanism is involved in ensuring the reorganization of the nodes of Ranvier after they've been disorganized?
That's a really great question, and I would recommend you look up Matt Rasband. So Matthew Rasband in Texas, he has spent his whole career doing beautiful work trying to understand how nodes are formed and disorganized and reformed. I don't have all the details. I know it has to be an interaction between the myelin and the axons. You can't have just one or the other, and you need to have the myelin actually functional to get to those mature perinodal loops to help segregate the Caspar in the right place in the sodium and the potassium channels. So it's not an area in which I have a lot of expertise, but I would recommend Matt Rasband, Rasband, to look up his work. He has amazing work in that area.
Okay, fantastic. Here's a nice question. Great talk. Agreed. Is the increased risk for cardiometabolic disease due to NASH or loss of innervation to the heart and pancreas themselves? And I might add to that a bit too to say, is the long-term persistent inflammation, is that linked directly to that initial neuronal injury, or is it the systemic complications associated with it, etc.?
Yeah, so in our model where we looked at NASH, we were doing a thoracic injury, so the heart innervation was not affected. That was all still intact above the level of the injury. We do know, however, below the injury, the sympathetic nervous system becomes overactive. So there's a lot of brain signaling that comes down the spinal cord normally to inhibit the sympathetics. Otherwise, we'd be in fight or flight all the time, right? So the sympathetics are normally controlled. When there's an injury, that break from the brain is lost. So sympathetic signaling is exacerbated. And we have some studies in the lab showing that if we can just remove the sympathetic fibers from the liver itself. And if we just do that, the inflammation in the liver is reduced and some of the effects of like insulin resistance are reduced. So I think that it's due to the injury causing a loss of descending inhibition on the sympathetics, and that sympathetic overdrive is causing inflammation in the liver. Also, the adipose, probably the pancreas hasn't been looked at that much, probably the kidneys. There's a lot of inflammation that's happening. Another problem I didn't even mention, however, is that also could be involved is gut dysbiosis. So there's work by Phil Popovich's lab and some others to show injury in rodents, and it's shown in humans as well, causes gut dysbiosis, but it also causes bacterial translocation. And it turns out the GI tract has a massive vein called the portal vein. Everything that comes out of the GI tract goes into the portal vein, and its first stop is the liver, because all of our nutrients, are processed and proceed through the liver before they go everywhere else. So a lot of bacteria that leave the GI tract end up in the liver, and the liver has receptors for that, and it drives inflammation also in the liver. So I think there are multiple mechanisms driving the systemic pathology that are going to take, it's not going to be an easy fix.
Right.
Yeah,
It's a multi-system problem, effectively.
yeah, right.
Great answer. Do OPCs also respond to GABA glycine to regulate myelination of inhibitory axons?
Yeah, there's some data to show they have GABA receptors and they can respond to GABA, and that might play a role in myelination. I don't know about glycine. They probably have glycine receptors. They have serotonin receptors. So these OPCs are really a fascinating cell population because if you think about it, like our brain and spinal cord have OPCs everywhere. They're throughout the white matter and gray matter, and we don't typically have a lot of demyelination, right? So why is the CNS investing in maintaining this large population of cells? What are they doing? So it's thought that they probably do a lot more than just sit around and wait to make an oligodendrocyte. They seem to be listening. They integrate in the hippocampus. They've been shown to actually get synapses from hippocampal neurons. So a neuron will synapse on an OPC, and then right next to it synapse on a dendrite. We know the OPC, when the neuron fires, OPCs are listening into that. So they seem to be integrating into circuitry, and we don't know exactly how or why. One thing is thought to be have to do with learning. So a lot of our axons in the adult CNS are actually not myelinated yet. So the corpus callosum is mostly unmyelinated. But as you learn new skills, like there's data showing if you learn to juggle or you learn a new foreign language as an adult, you'll increase the amount of myelination within the brain in specific areas. So it's thought that they are listening to these neurotransmitters, listening to changes in the circuitry, and somehow figuring out when and where they need to make a new oligodendrocyte. They're probably doing more things than that too, but that's one of
Right.
the main things that we know.
Right. Okay, wonderful. Um, to what extent is the liver steatosis related to energy use in SCI or
Hmm.
due to disuse?
Right. So that's probably does contribute. So we know that people, if they're in a wheelchair, they're going to have less physical activity compared to people who are not in a wheelchair. But there is data to show this steatosis in our animal models and increased adipose in humans happens very rapidly. So it happens like our animals show within a week, right? So they're not it's hard to imagine that it's just due to inactivity. Um, and also there's data that looked at Paralympics. So people who are, in these Paralympic games, much more active, even I'm sure than I am, right? They still tend to have some of these same issues. So it's probably partly due to inactivity, but then also we think there are active mechanisms that are driving this, the steatosis and some of the metabolic problems in this population.
Okay, wonderful. I think maybe we'll try and squeeze in one last
All
one,
right.
if we can. Have you used NG2DS red mice in your spinal cord injury work, and if so, what value might that model have?
We have not used the DS red mice. One of our mice models actually was from Akiko Nishiyama, who made the DS red mice. But no, I have mice that the NG2 cells express GFP on their membranes, and they label OPC processes beautifully. So we've just stuck with that for some of our reporter studies.
Okay, wonderful. Unfortunately, I think we have to wrap it up there. I think we go on for much longer. Um, Dana, I really want to thank you for sharing your insights today. It's been absolutely a pleasure having you with us.
Thank you so much. Thank you for the invitation, and thanks to everybody for listening. Reach out if you have any other questions.
And a final thank you to our partners for helping make this series possible, particularly the Society for Neuroscience, the today's host society, and today's sponsor, Alzet, as well. In closing, we hope you enjoyed this Inside Scientific webinar, and we hope to see you again next time. Thank you, everyone.